When we study glucose metabolism, our minds naturally jump straight to glycolysis and burning sugar for ATP. However, your cells operate a massive parallel highway that cares nothing about making energy. Welcome to the Pentose Phosphate Pathway, also known as the anabolic shunt. This critical pathway serves as a cellular logic circuit, balancing a cell's immediate survival demands with its long-term growth imperatives. While this pathway originates directly from the oxidation of glucose, its primary role inside the cell is distinctly anabolic. Operating completely parallel to glycolysis, it functions as a hexose monophosphate shunt. Instead of harvesting carbon to generate ATP, it diverts substrate to accomplish two primary goals: generating reducing equivalents in the form of NADPH for cellular defense, and manufacturing 5-carbon sugars required for nucleotide synthesis. Where exactly does this take place? At the cellular level, the entire pathway is localized strictly within the cytosol, the fluid cytoplasm of mammalian cells. Because of its specialized anabolic roles, the pathway's activity is highly pronounced in tissues with intense biosynthetic or oxidative demands. On an organism level, this makes it highly active in the liver for fatty acid synthesis, the adrenal cortex for steroid hormone synthesis, and mammary glands during lactation. In plants, it is prominently active within the plastids. The entire pathway is built on a bipartite architecture divided into two completely opposite phases, the oxidative phase and the non-oxidative phase. The oxidative phase is a strictly irreversible one-way chemical street engineered to generate high-grade NADPH reducing power. Conversely, the non-oxidative phase is a highly reversible structural engine. It uses trans-ketolase and trans-aldolase enzymes to shuffle sugar-phosphate-carbon backbones, tailoring outputs like ribose 5-phosphate to match what the cell needs at any given moment. Let us walk through the irreversible oxidative phase map. In step 1, G6P passes through the critical G6PDH toll booth, yielding our very first molecule of reducing power as NADPH. In step 2, a lactinase enzyme facilitates rapid hydrolysis, opening up the sugar's closed ring structure. Finally, step 3 drives a definitive oxidative decarboxylation. This strips away electrons to yield a second molecule of NADPH, kicks off a molecule of carbon dioxide waste, and concludes the phase by leaving us with a 5-carbon sugar, ribose 5-phosphate. Once we harvest our raw reducing power, the 5-carbon intermediates enter the fluid, highly flexible, non-oxidative phase map. During initialization, Ru5P is isomerized into ribose 5-phosphate, the foundational building block for downstream growth. From here, a highly dynamic interconversion engine powered by trans-ketolase and trans-aldolase flexibly rearranges carbon units between various 5, 7, and 4-carbon sugars. If the cell does not require these sugars for growth, the excess intermediates are seamlessly rerouted right back into carbohydrate metabolism through fructose 6-phosphate and glyceraldehyde 3-phosphate. To understand the precise chemical accounting of this pathway, let us look at its net stoichiometry. The primary balanced equation dictates that for every 3 molecules of glucose 6-phosphate and 6 NADP+ entering the system, the pathway yields 3 molecules of ribose 5-phosphate, 3 molecules of carbon dioxide waste, and 6 molecules of highly valuable NADPH. If the cell recycles the carbon backbones completely through the non-oxidative engine, those 3 ribose 5-phosphates are rearranged into exactly 2 molecules of fructose 6-phosphate and 1 molecule of glyceraldehyde 3-phosphate. Everything we have looked at supports the twin pillars of cellular output. The left pillar represents redox and biosynthesis. By generating NADPH, the pathway provides vital reducing equivalents required for maintaining cellular redox balance, neutralizing oxidative stress, and driving the heavy synthesis of fatty acids and cholesterol. The right pillar represents proliferation. It provides the essential 5-carbon sugar backbone required for nucleotide synthesis, acting as the critical precursor for DNA, RNA, ATP, and GTP. Let us look closer at how the cell survives toxic environments. Reactive oxygen species, or ROS, are natural but highly dangerous byproducts of routine cellular metabolism. To neutralize them, the cell relies on a potent protective scavenger called glutathione. However, the glutathione pool must be constantly recharged with electrons to stay functional. In this mechanism, NADPH acts as the ultimate electron donor, regenerating reduced glutathione so it can continually clear out ROS, preserve cellular integrity, and turn dangerous oxidants into harmless water. This defense system leads us to a profound biological reality known as the erythrocyte paradox. Mature red blood cells lack mitochondria entirely. This renders them completely incapable of generating ATP via oxidative phosphorylation or utilizing the TCA cycle. Because they lack these alternative metabolic hubs, the pentose phosphate pathway is the absolute solitary source of NADPH for red blood cells. Without it efficiently maintaining the reduced glutathione pool, erythrocytes suffer rapid, unchecked oxidative damage, leading to severe cell membrane hemolysis and premature destruction. To ensure resources are never wasted, the pathway utilizes a highly responsive internal feedback loop dictated entirely by the cellular ratio of NADP+ to NADPH. When the cellular NADPH concentration climbs high, it indicates the cell's antioxidant and biosynthetic demands are fully satisfied. Mechanistically, these high product levels act as a direct feedback inhibitor, clamping down on the primary gatekeeper enzyme, G6-PDH. This effectively shuts down the entry gate. The moment cellular NADPH levels drop, this inhibition lifts, and the metabolic circuit automatically re-engages. Beyond internal automation, the pathway responds to external orchestration. Hormonally, insulin stimulates glucose absorption and directly upregulates G6-PDH expression to accelerate the pathway, while glucagon suppresses it. At the genetic transcriptional level, severe oxidative stress triggers the transcription factor NRF2. NRF2 migrates to the DNA to radically increase the expression of downstream PPP enzymes, scaling up defense exactly when a threat arrives. Finally, substrate routing plays a role. A high local concentration of G6P physically pushes raw material directly into the shunt. Ultimately, the pentose phosphate pathway is far more than just a minor detour or an alternative route for processing glucose. It acts as a central metabolic logic circuit regulating cellular life. By sitting precisely at the crossroads of carbohydrate metabolism, it grants the cell absolute metabolic flexibility. It dynamically balances the cell's two most fundamental competing imperatives: protecting itself from immediate biochemical destruction today, and building the structural molecular foundation to grow and replicate tomorrow.